Hello everyone. This time we are diving into a topic that's literally happening inside you right now, every single second. Imagine you're walking through a busy market. It's vibrant and full of energy, right? But sometimes a bit chaotic. A loose wire here, a small spark there, harmless mostly. But if left unchecked, that spark, that little spark, can turn into a raging fire, causing havoc. Now, shrink that image down, way down, into the microscopic world of your cells. What is that spark? That's what we are discussing today. It is the free radical. And the firefighters that are desperately trying to keep the peace, they are the antioxidants. So we are going to discuss about these two opposite forces, the free radicals and the antioxidants. And why is this important for you? Because there's a very delicate balance that exists between these two. This is the oxidative stress. It is not just a fancy biochemical term. It's a major player in the pathogenesis of everything from aging, heart disease, diabetes, cancer, etc. It helps you understand the disease and guide the patient in a better way. We start with the free radical. At their core, free radicals are simply molecules with an unpaired electron in their outermost orbital. Think of it like a person who lost their wallet at a wedding. They are agitated, desperate, and will borrow or steal from anyone nearby to feel complete. This electron theft is what makes them so reactive and damaging. The first question arises, how are these actually formed? Now, the fact here is that our bodies are continuously producing free radicals with every breath that you take. And the biggest culprit is actually mitochondrial respiration. So, mitochondria, as you might know, that they are the power station of your entire body. They are constantly generating ATP through the electron transport chain. But this process isn't 100% efficient. About 1 to 2% of oxygen gets incompletely reduced, especially at complex 1 and 3. Here, oxygen forms superoxide. This is the superoxide radical, and this is the first and the most common and relatively mild free radical. Think of it as a small, manageable spark. Now, other than this, there are several enzymatic reactions which deliberately produce free radicals. Now, the few examples of these include NADPH oxidases, or in short, NOx. In our immune cells, like neutrophils, macrophages, these NOx enzymes are like a special forces unit. They deliberately produce massive amounts of superoxide radicals during the respiratory burst to kill bacteria and fungi. This is a very controlled use of free radicals for defense, like controlled exposure to clear a path. Then there are xanthine oxidases. These are involved in purine metabolism and they can generate superoxide radicals as well as peroxide radicals, hydrogen peroxide. They are really elevated in conditions like reperfusion injury. Another example is the nitric oxide synthase, or NOS. These produce nitric oxide radicals. Nitric oxide is a vital signaling molecule for vasodilation, but it's also a free radical. It's useful in one way but harmful in another. Then there are various exogenous sources. We also call these the environmental invaders. Examples of these include pollution. Think of the smog over Delhi or Mumbai, but the particulate matter, ozone, nitrogen oxides, they all trigger free radical formation. Then there is UV radiation, too much sun exposure, especially harsh Indian sun on your skin. Then there is smoking or cigarette smoke, also alcohol, certain drugs. These are common culprits. And then there are ionizing radiations. These include X rays or gamma rays. This is how the free radical is formed. Once it forms, during the formation stage, it's just like a small spark. It moves to the next stage, which is the chain reaction, when the spark transforms into an inferno. So after the formation, free radicals form even more dangerous radicals. For example, superoxide gets converted to a hydrogen peroxide. Now, the special point about hydrogen peroxide is that it doesn't have an unpaired electron, but it is highly reactive and can easily diffuse across membranes, making it dangerous. Then there is the hydroxyl radical. This is the most dangerous radical. It's formed from H2O2, especially in the presence of transition metals like iron or copper. There is a special reaction which we study about, and that is the Fenton reaction, where Fe2 plus and H2O2 react to form Fe3 plus and OH. Now, this is also one of the reasons why, in certain conditions where there is ion overload, like hemochromatosis or repeated transfusions, where there is excessive ion which fuels hydroxyl radical formation. And this hydroxyl radical is dangerous because it reacts with almost anything it touches, causing widespread damage. Okay, so this is how the chain reaction occurs. Now, the first one was formation, then there was the chain reaction. Then we come to the third one, which is molecular damage. Molecular damage is the aftermath. Once formed, these radicals wreak havoc on vital cellular components. This is what we are most afraid of. In this, the most initial kind of molecular damage is lipid peroxidation. So they attack polyunsaturated fatty acids, or PUFAs. Now, these PUFAs are present in cell membranes. So what this does is, this is like punching holes in a balloon, which leads to loss of membrane integrity, and the fluidity of the membrane changes and cells become more leaky, and various byproducts are formed. One of the common byproduct is malondialdehyde, or in short MDA, which is actually used as a measurement as it acts like a marker of oxidative stress. The next one is protein oxidation. Now, free radicals modify amino acids, leading to protein fragmentation, followed by aggregation and then loss of enzyme activity. It completely rusts the vital machinery. Then there is DNA damage. So, these free radicals can break the DNA strands. They can even modify the bases and cause mutations. And they can increase the risk of cancer by affecting gene expression. In short, they can completely corrupt the blueprint of the cell. Okay, so this is in short about the various molecular damage which can occur. So we have discussed how they are formed, how the chain reaction can occur, and then we have seen the molecular damage that it can inflict. Now let's move on to antioxidants. These are basically our internal fire brigade system. Now, fortunately for us, our cells aren't helpless. We have sophisticated multi-layered defense systems. This is provided by antioxidants. And the first type of antioxidants are enzymatic. There are various enzymatic reactions to neutralize the radicals. The first enzymatic reaction we see is that of superoxide dismutase. It quickly converts the superoxide radical to H2O2. And there are different versions of superoxide in our body. The one in the mitochondria is associated with manganese, and the one in the cytosol is associated with copper and zinc. The next one is catalase. This enzyme is like the big gun for H2O2. It is found mainly in the peroxisomes, and it rapidly breaks down H2O2 into water and oxygen. Now, a special fact here is, if you have ever seen H2O2 bubbling on a wound, that's your catalase working. The next one is glutathione peroxidase. This is a selenium containing enzyme, and it reduces H2O2 and other hydroperoxides using glutathione. Then there is glutathione reductase. This enzyme is crucial for recycling our antioxidants. It regenerates the active reduced form of glutathione, which is GSH, from its oxidized form, which is GSSG. So it's basically doing the opposite of this reaction. Now, this reaction is actually where the pentose phosphate pathway comes in. There's an enzyme called glucose 6-phosphate dehydrogenase enzyme, which is key for producing NADPH. And if you have G6PD deficiency, which is common in some Indian populations, you can't make enough NADPH. So you can't regenerate GSH effectively. So this reaction can get hampered. Now, why this is relevant is because if there is deficiency and you can't regenerate GSH, the RBCs become vulnerable to oxidative stress, which leads to hemolytic anemia. And whenever we study such a case, such hemolytic crisis is triggered by certain specific drugs like primaquine or sulfa drugs. Then there is the thioredoxin system, which is another important redox system, which also uses NADPH for reducing disulfide bonds and protecting against stress. We consider all these enzymatic reactions as enzymatic antioxidants. Now that we have seen the enzymatic antioxidants, let's also see the non-enzymatic ones. All these non-enzymatic ones are actually the scavengers. They are molecules that directly neutralize free radicals, often from our diet. Now, examples of these are water soluble, which include vitamin C, glutathione, uric acid, these three. Vitamin C acts as a powerful electron donor and it can neutralize various free radicals. Think of it as a quick relief worker. It also helps regenerate vitamin E. So it has a very significant role. Then we have glutathione, which is our most abundant intracellular non-protein thiol. It is a tripeptide, glycine, cysteine, and glutamate, and it directly detoxifies radicals along with heavy metals and xenobiotics. Then uric acid is a byproduct of purine metabolism and it's a potent antioxidant in plasma. Then the lipid soluble examples. This is vitamin E, coenzyme Q10, carotenoids, etc. So this vitamin E or alpha tocopherol, this is the primary fat soluble antioxidant and it is embedded in cell membranes. It acts like a shield protecting membranes from lipid peroxidation. Then the coenzyme Q10 is also called ubiquinone. It is found in mitochondria and it is part of the electron transport chain and it is also a potent antioxidant. Then we have carotenoids. These include beta carotene, lycopene. They are found in colorful fruits and vegetables, carrots, tomatoes, mangoes. This is only a very small list of non-enzymatic antioxidants, but these are pretty important ones. Next, we move on to the antioxidant cycle. Now, these systems aren't isolated, they work in a beautiful synchronized fashion. Imagine, our, imagine a football team. Each player has a role, but they support each other. Like vitamin E supports the membranes, like it's acting like a shield, but then gets oxidized. So, vitamin C comes and regenerates it. And as this is being depleted, glutathione regenerates vitamin C. And as this is getting degraded, NADPH from the pentose phosphate pathway regenerates the glutathione. Now, this continuous recycling is key to sustain defense. It's why a balanced diet with a variety of fruits and vegetables is so vital and not just isolated supplements. Now, how is everything regulated and controlled? So, how does the cell know when to unleash its antioxidant army? Now, here we can talk about the NRF2 KEAP1 pathway. This is the master orchestrator or coordinator of our antioxidant system. Now, in normal times, this NRF2, which is nuclear factor erythroid 2 related factor, is held captive in the cytoplasm by KEAP or KEAP1. KEAP1 acts as a sensor and it also tags NRF2 for destruction. Now, this keeps its levels low. Now, when there is oxidative stress, when the reactive oxygen species or electrophiles increase, they modify specific cysteine residues on KAP1. Now, due to the modification of these residues, the KAP1 kind of gets stunned and it doesn't perform its role that well. And what happens is that the NRF2 increases. It takes charge. Now, this free NRF2 quickly moves to the nucleus, and what it does is it binds to specific DNA sequences called antioxidant response elements, or ARES. This binding activates the transcription of a whole battery of antioxidant genes. So, in a way, once the antioxidant genes are stimulated and the antioxidant enzymes get activated, an antioxidant reaction takes place in the body and the body fights against oxidative stress. So, it's an ingenious system which allows our cells to dynamically upregulate the defenses when needed, much like a rapid deployment force responding to a crisis. Now that we have delved into deep biochemical aspects of free radicals and antioxidants, let's talk about a couple of clinical correlations. The first one I want to discuss is the G6PD deficiency. Now, you'll encounter patients, especially from certain ethnic groups in India, who develop hemolytic anemia when given oxidative drugs, example antimalarials like primaquine or self-antibiotics. So the drug history and the family history can be very valuable. The next one is diabetes mellitus. So chronic hyperglycemia leads to increased reactive oxygen species production, which contributes to microvascular retinopathy, neuropathy, and nephropathy, and other macrovascular complications. Oxidative stress damages endothelial cells, impairs insulin signaling, and it promotes inflammation. Then there is cardiovascular disease. The oxidation of LDL cholesterol is a critical initial step in the formation of atherosclerotic plaque. Now, our body is trying to prevent this with the help of vitamin E and other antioxidants. Fourth one is neurodegenerative diseases. So these include Alzheimer's and Parkinson's disease. They all show significant oxidative damage in the brain, and it appears that the brain is particularly vulnerable to high oxygen consumption and high lipid content, and so it is susceptible to lipid peroxidation. Then there is cancer. Chronic oxidative stress can cause DNA mutations, which can promote carcinogenesis. And some cancer therapies induce oxidative stress to kill cancer cells. It's a complex relationship. Then finally, air pollution and lung disease. So in highly polluted cities, our lungs are exposed to constant oxidative stress. This contributes to asthma, COPD, and lung cancer. Now, I would like to conclude this topic here, but I want to give you a high yield summary. So, we learned what free radicals really are. They are molecules with unpaired electrons, and they are highly reactive, and they cause damage to lipids along with proteins and DNA. They are primarily formed in mitochondrial electron transport chain and also various enzymes like NOx and external factors like pollution. Then we learned about oxidative stress, which is the imbalance when free radical production overwhelms antioxidant defenses. We learned about the key free radicals. These include superoxide, hydrogen peroxide, and the most dangerous hydroxyl radical. We learned about the Fenton reaction. Then we learned about antioxidant defenses. These were enzymatic and non-enzymatic. In enzymatic, we saw SOD, Catalase, glutathione peroxidase, glutathione reductase, etc. And we learned about G6PD. Then we talked about non-enzymatic antioxidants like vitamin C, E, ubiquinone, etc. Then we talked about the regulation, which was controlled by the NRF2 KEAP1 pathway, which is an ingenious pathway. Then we saw a lot of clinical relevance, which is oxidative stress contributing to aging, neurodegenerative diseases, cardiovascular diseases, diabetes, and cancer. So that concludes our topic on free radicals and antioxidants. We have got a broad overview of what this topic is all about. There are still lots and lots of examples that can be studied in this. Examples of free radicals, oxidants, and antioxidants. You can find a lot of these examples in most of the biochemistry books. But once you know the concept of how it all works, you can understand it in a better way. Hope this gives you a good overview and intro into this topic, and I hope to see you soon in the next video. Thank you for watching.